Good morning, everyone. Today, we are going to focus on essential aspect of your A-level studies, mastering general insurance through multiple choice questions. By the way, don't forget to hit the like button, turn on your notification to stay updated, and subscribe to Jom Study on YouTube channel. Without further ado, let's get into it. A student will note describing photomicrograph of 4 cells, cells 1, cell 2, cell 3, and cell 4. Which table identify the type of cells and the type of microscope used to take each photograph? The answer will be C. The diagram shows a slide of a transverse section of a stem. This diagram is the same size as the actual slide. A student observed this slide using a light microscope at a magnification of multiply 40. The student made a plan drawing of a stem which was 20 centimeters in diameter. The student labeled the plan transverse sections of a stem multiply 40, and which statement explain why this label is not correct. The answer is the image size in the drawing was larger than multiply 40. Which cell structures may contain systema are flattened membrane-bound sets of fold found in various cell structures. Based on this, the correct answer will be B, which can be explained. Chloroplasts and mitochondria contain structures similar to cisterna. In chloroplasts, they are found in the thylakoid membranes, and in mitochondria, they are present in the inner membranes fold called cister. So the endoplasmic reticulum and Golgi body do not contain cisterna in the same sense. Which row correctly describe the function of the cell structure? The row that correctly describe the function of the cell structure will be D, which is lysosomes, spherical sacs containing hydrolytic enzyme. Mitochondria is ATP synthesis, and smooth endoplasmic reticulum, where the functions is related to glycoprotein productions, and the Golgi body is a lipid production. Number 5 is a common exam question which is already exists in November 2023, which the diagram show 3 circles, 1, 3, and 5, and share structures 2 and 4. We should correctly identify the 3 circles and some of the structures that are shared between them. So, prokaryotes are single-cell organisms that lack nucleus and membrane-bound organelle. They typically have circular DNA, which is found in the nucleate region. Mitochondria and chloroplasts are organelles found in eukaryotic cells, and they are believed to have originated from prokaryote placenta through endosymbiosis. And both mitochondria and chloroplasts have their own circular DNA, which is similar to prokaryotes and replicate independently within the eukaryotic cells. Which comparison of bacterial cells wall and plant cell wall is correct? The correct comparisons of bacterial cell wall and plant cell walls is D, which is bacterial cell wall, which is made of peptidoglycan, and for the plant cell wall, is made of polymer of beta-glucose. A glycosidic bond is broken and 2 monosaccharides are formed during a positive test for a non-reducing sugar, which will identify the catalyst and reactant in this process. So the correct answer, which row that identify the catalyst and reactant in this process is B, which catalyst is hydrochloric acid and reactant is a sucrose and water. In this context, hydrochloric acid acts as a catalyst in the hydrolysis reaction, which break down sucrose into its monosaccharide component, fructose and glucose. Which pair of statements are correct? So B is the correct answer because glycolipids are present in all cell membrane, and plants store carbohydrate as a starch. And which is the general formula for cellulose? The answer will be C8H10 and O5 in bracket is N. Cellulose is a polysaccharide made of a repeating glucosinolate, but it does not have the hydrogen and oxygen in the same 1:1 as glucose. And the correct formula reflects the monomeric unit of cellulose, which is glucose with one water molecule. Remove the linkage between units. A triglyceride consists of glycerol and three different fatty acids: linoleic acid, oleic acid, and palmitic acid. And the diagram shows one possible arrangements of the fatty acids L, O, and P in the molecule. And what is the total number of different arrangement of the fatty acids in this triglyceride? The answer will be 3. Which statement about a peptide bond are correct? The correct statements about a peptide bonds are: it contains 4 different atoms, which is carbon, oxygen, hydrogen, and nitrogen. And it is important in the primary structure of protein as it links amino acid together. So the answer will be 2 and 4. Which statement about the primary structure of a proteins are correct? The correct statements about the primary structure of the protein is: it is determined by the sequence of DNA bases, and it is unique to the protein, and it determines the tertiary structures of the protein. And the primary structure refers to the sequence of amino acid in a protein, which is specified by the DNA sequence. And this sequence is unique to each protein and influences how the protein will fold into its tertiary structure. A mutation occurs within the DNA sequence coding for an enzyme, causing a decrease in the rate of a reaction catalyzed by this enzyme. Which statements could explain the decrease in the rate of reactions? The answer will be: the active sites of the enzyme might have changed shape, and so it is no longer complementary. And this could be reduce the enzyme ability to bind to the substrate effectively. And the activation energy for the reactions with the mutated enzyme is greater than with the non-mutated enzyme. Instead, because it's described an external factors, because this would mean the enzyme is less efficient in catalyzing the reaction. So the statement is considered not necessary because to the mutation directly affecting the enzyme structures. Instead, it describes an external factors inhibitor affecting the enzyme activities. Which row is correct for an enzyme with a low Michel's-Menten constant? The answer will be B, because affinity of enzyme for substrate is supposed to be high because a low Km indicates that the enzyme bind the substrate more effectively, higher affinity. But when it gets to substrate concentrations at minimum reaction rate, it's supposed to be low because a low Km means that the enzyme reaches its maximum reaction rate at the lower substrate concentration. 15. The diagram shows a glycoprotein embedded in the cell surface membrane of a human red blood cells. This glycoprotein is part of the system of cell surface blood group recognition sites. And which row identify the roles of these glycoproteins and region P and Q of the molecule? The answer will be A. The diagram shows the transport of ions across the cell surface membrane. Inside the cells, there is a low concentrations of sodium ion and a high concentrations of potassium ion. And outside the cells, there is a low concentrations of sodium ions and a high concentrations of potassium ion. The carrier molecule is a pump which exchanges sodium ions for potassium ion, and which ionic movements are represented by the arrow. The answer will be A. 17. The indicator crisis rate changes from red to yellow when put into an acid. Some blocks of agar-containing crisis rate were cut to different sizes and put in acid. All other variables were kept constant. The blocks were measured in mm. Which block became completely yellow most quickly? So to determine which block of agar containing crescent rate become completely yellow most quickly, we need to consider how the size of the block affect the rate at which the acid can diffuse into agar. And when an acid is added to the agar block, it diffuses from the surface into the interior. The larger the surface area relative to the volume, the faster the diffusions of the acid can occur, which means the block will change color more quickly. For example, for the block A, surface area, we get the value of 2,700 millimeter cube. And for the surface area to volume ratio, we get 0.8. And for block B, which is B, 6 multiplied by 6 multiplied by 6 millimeter, where the surface area is equivalent to 216 millimeter cube, and the surface area to volume, we get the value of 1. And for the block C, which is 6 multiplied by 12 multiplied by 12, we get the surface area is equivalent to 576. We get the 864 millimeter cube. And for surface area to volume ratio, we get the value of 0.67. And for the block D, we get the surface area about 1,728 millimeter cube. And for the surface area to volume ratio, we get 0.55. The block with the highest surface area to volume ratio will be the one that becomes completely yellow most quickly because the acid can diffuse more efficiently into it. So the answer for this will be B. 6 multiplied by 6 multiplied by 6 has the highest surface area to volume ratio and completely yellow most quickly. When red blood cells are put into pure water, they burst hemolysis. Which statements explain this hemolysis? Statement 2 and 4. Statement 2 is the cell surface membrane of red blood cells are not supported by cell wall. And number 4, water enters the red blood cells by osmosis but does not leave the cells. And number 19, how many copies of each DNA molecule will be found in a cell at each stage of the mitotic cell cycle? The answer will be A, because G1 of interphase is a cell has 1 copy of the DNA molecule. And this is the stage before the DNA replication occur. But cytokinesis is after mitosis, which is M phase. During cytokinesis, the cells split into 2 daughter cells. And each daughter cells will have 1 copy of the DNA molecule, assuming the division was successful. So in G1 of interphase, there is only 1 copy of each DNA molecule. But at cytokinesis, each of the 2 daughter cells will have 1 copy of each DNA molecule. So the correct answer will be A. Which metabolic processes will be very active in the cells that have just completed cytokinesis? There will be ATP formation, and the other one will be protein synthesis. Why? ATP formation is a cell which needs energy to support various activities and to prepare for the next round of the cell division. And whereas for DNA replications, and this process will begin shortly after cytokinesis, as the cell prepare for the next cell cycle phases, which call it, or also so-called as S phase. And on the other hand, protein synthesis is also active as the cells build new proteins which are required for the growth and the function. A mutation occurs in a gene which prevents the production of telomerase. What is the effect of this mutation if it is occurred in the bone marrow stem cells? So the answer will be bone marrow stem cells eventually no longer divide. This can be explained if a mutation occurs in a gene that prevents the production of telomerase in bone marrow stem cell. The effect is likely to be bone marrow stem cells eventually no longer divide. Telomerase is an enzyme that maintains the length of telomerase at the end of chromosome. In a stem cell, telomerase activity is crucial for maintaining the ability to divide and produce new cells. Without telomerase, the telomeres gradually shorten with each cell division, eventually leading to cellular senescence or death, and this would result in the bone marrow stem cells, losing their ability to divide, which in turn impact the productions of the blood cells. What are the products when a DNA molecule replicates? The answer will be 2 molecules of DNA, each made of a paired sequence of nucleotides. This can be explained. Each new DNA molecule consists of 2 strands, and each strand is composed of a sequence of nucleotides that pair with the complementary nucleotides, and on the other hand, on the other strand, forming the characteristic double helix structure of DNA. Following translation, the alpha polypeptide chain of hemoglobin, alpha globin, undergoes modification. During this modification, the first amino acid is removed, leaving 141 amino acid residue. How many nucleotides does the gene coding for alpha globin contain? So to determine how many nucleotides the gene coding for the alpha globin contains, we need to account for the fact that each amino acid in the protein is encoded by a set of 3 nucleotides, a codon. Since the alpha globin protein after translation has 141 amino acids, with the first amino acid being removed, the gene must code for 141 amino acids plus an additional 3 nucleotides for the stop codon. So, the number of nucleotides required is 141 amino acids multiplied by 3 nucleotides, and then you have to divide it by nucleotides or amino acid, and you need to plus 3 nucleotides, is 3 nucleotides for the stop codon, where you get the value of 426 nucleotides. So, the gene coding for alpha globin contain 426 nucleotides. Which statement correctly describes transport pathway in dicotyledonous plant? The answer will be an A. Why is an A? In the apoplast pathway, water does not move through plasmodesmata. In the cyclus pathway, water does not move through intercellular spaces, and the apoplast pathway may be blocked by the Casparian strip. And the photomicrograph show a transverse sections through a root. What is the simplest ratio of the diameter of the root, excluding root hair, to the diameter of the vascular tissue and endodermis? The answer will be 40:11. Which property of water molecules are important in the outward flow of water through xylem? The answer will be water molecules are attracted to each other by hydrogen bonding. Water molecules are attracted to cellulose by action. And water molecules have high cohesion in water columns. And these properties contribute to the cohesion tension theory, where hydrogen bonding and cohesion create a continuous water column, while action to the xylem walls helps to counteract gravity and facilitate outward movement. What is correct for a volume-shifting element that is unloading to a sink? The answer will be, when uploading occurs, the volume-shifting element typically has a more negative water potential compared to the sink. And this is because the unloading process involves the removal of solute from the volume, which causes the water potential in the shifting elements to become more negative relative to the sink. And in terms of the lignifications of the cell's wall, shifting elements do not have lignified cell wall, which is absent. Lignification is generally absent in the shifting elements, whether they are loading or unloading. So lignification is more characteristic of xylem cells. The answer will be C. And which statement concerning transpiration is correct? The answer will be water arriving at the spongy mesophyll cells via the simplest pathway must move by the osmosis through the cell surface membrane before evaporations from the surface of the cells. And this can be explained when water moves into the spongy mesophyll cells by the simplest pathway and then must go through the cell membrane by osmosis before it can evaporate into the air spaces. And then 29, the diagram shows a section through the heart and the associated with the blood vessels. Number 1 is represent pulmonary artery. 2 is left atrium. 3 is the right atrium. And 4 is a right ventricle. What is the correct for the flow of blood through the heart? Deoxygenated blood returns from the body to the right atrium, which is 3. Right atriums via the superior and inferior vena cava, and the right atrium contracts, sending blood through the tricuspid valve into the right ventricle. And the right ventricle then pumps the blood through the pulmonary valve into the pulmonary artery, and then to the lung for the oxygenation. Oxygenated blood returns from the lungs to the left atrium via the pulmonary veins, and the left atrium contracts, sending blood through the mitral valve into the left ventricle. And the left ventricle then pumps the oxygenated blood through the aortic valve into the aorta and throughout the body. So the answer will be 3, 4, 1, and 2. Blood tissue fluid and limb each have a different composition, and which will show the composition of limb. Limb has a composition that is relatively similar to tissue fluid, but has fewer proteins and more lymphocytes compared to tissue fluid. And it also generally has less lipid compared to chyle, which is limb from the digestive system. And the composition of limb typically include water, antibodies, and lipid, supposed to be all present. And therefore, the correct row showing the composition of limb is A. Which row show the change in concentration of some substances in red blood cells when carbon dioxide diffuses from active cells? So when carbon dioxide diffuses from active cells into the red blood cells, several changes occur, which is carbonic hydrolysis. The enzyme catalyzes the conversion of carbon dioxide and water into carbonic acid, when then this associates into bicarbonate, hydrogen carbonate, ions, and hydrogen ion. And the activity of carbonate hydrolysis typically increases the response to higher carbon dioxide level. And hydrogen carbonate ions is converted into carbonic acid, which deassociate into bicarbonate ion and hydrogen ion. So the concentration of bicarbonate ion increases in the red blood cells. And finally, we have hydrogen ion. What will happen to hydrogen ion? The disassociation of carbonic acid produces hydrogen ion, so their concentration also increases. Based on these three changes, the correct row showing the changes in the concentration of some substances in red blood cells when hydrogen carbonate ions diffuses from active cells is supposed to be B. The diagram shows the pressure changes in various structures on the left side of the heart during the cardiac cycle. At the end of this period, which period is the ventricle force of the blood? So the answer will be A. The photomicrograph shows a cross-section through the lining of the part of the respiratory system. Which statements are correct? Smooth muscle is visible, and the section cannot be from a bronchial as cartilage is visible. In the lung, oxygen and carbon dioxide pass through cell surface membrane by diffusion. Which row is correct? The answer will be D, which is oxygen from the air need to pass through about 5 membranes, which is including the alveolar and capillary walls to get into the blood, and the carbon dioxide need to pass through 4 or 5 membranes to move from the blood into the air. Which flow diagram correctly describes the effect of tar entering the lungs? There will be carcinogens come into contact with DNA, and then goes to this happen, mutation occurs, and then it leads to uncontrolled cell division. Which list contain only infectious diseases? The answer will be C, HIV, AIDS, malaria, measles, and TB. And which disease is caused by eukaryotes? It can be caused, the disease that caused by the eukaryotes is malaria. Malaria is caused by Plasmodium species, which are protozoan eukaryotes, and the other diseases listed are caused by bacteria or viruses which are not eukaryotes. A vaccine is available against most common strain of the influenza virus. The virus that causes influenza often undergoes mutation in the gene coding for its antigen protein, which statement explains why vaccinated people are not immune to a mutated influenza virus. The answer will be they will not have a secondary immune response to the mutated antigen, and their memory cells do not recognize the mutated antigen. That will be the answer for why vaccinated people are not immune to a mutated influenza viruses. And 39, which statement correctly describe lymphocytes? Here is some of the statements about lymphocyte. First, each B lymphocyte produces a specific types of antibody rather than several types. That is number 1. Number 2, some B lymphocyte and T lymphocyte become memory cells, and both B lymphocytes and T lymphocyte can become memory cells. And number 3, plasma cells secrete antibody into the blood plasma. And number 4, some T lymphocytes, specifically helper T cells, stimulate macrophages to enhance the ability to kill infected cells. So the correct information given is only number 2, 3, and 4. Addison disease can occur when antibodies are produced in response to an enzyme found in some organs of the body. Which statement correctly describes Addison diseases? The answer will be 1, 2, and 3. 1, it is a non-infection disease. It is a type of autoimmune disease, and antibodies are produced against a self-antigen. So all these 3 statements are accurate, and the correct answer will be A. That's all for today's presentation. Thank you very much for your cooperation, and hope you are doing very well in your examination. And please don't forget to hit the like button and turn on your notifications to stay updated, and subscribe to Jom Study Lab YouTube channel.